Elsa, thanks very much for joining us, and please, you can proceed with your presentation, right?
Okay, good morning, everyone.
Good morning.
And thank you so much for the invitation as a panelist for this important event. So I'm veterinary and I study about Leptospira. So that's my contribution, and I'm proud to obtain isolates from sylvatic rats and mice in Angola, and to explore another ways for diagnosis point of care that's needed in our country. So recently, I joined the CISA group in Cachito village to carry out a project, namely monitoring of the diarrhea in under 5 children in Bengo Province. That implicates different methods and the access to remote and poverty locations. So, children. So firstly, those were a challenge for me and for the interviews, but a new opportunity because few studies about intestinal microbes community were published. So there haven't consistent information about intestinal bacteria in that location and in Angola in general. Thus, our aim is to access the intestinal bacteria and the rotavirus genotypes among children under 5 in the main public hospital of Bengo Province. Bengo is a big province in Angola. So, and an important source of agriculture products. The people live in conditions of poverty and lack of sanitary conditions, and living predominantly in rural areas. So we are located in Cachito, is in middle of the map. So we have been collect water stools from children that attended the hospital after the permission from the caregiver. In the same time, we ask clinical and epidemiological questions. The samples will be processed under safe cabinet. That's our short protocol. Before we culture, we make the culture and incubate for 24 hours at 37 Celsius degrees. We transfer 1 or 2 colonies to one tube, to a couple of tubes, and dilute until 600, and transfer to VTEC cards. We use 2 VTEC cards, one for intestinal gram-negative bacteria and another for the test for sensibility of antibiotics. We use a potential machine, namely VTEC compact, for identification. For rotavirus, we screening by rapid tests. So when the test positive, we extract the RNA and we make the RNA transcriptase to obtain cDNA, and then we proceed the PCR into two ways: the normal PCR and the nested PCR, depend of the genotypes who that I want to genotyping. Our results shows that we can identify enterobacteria. We can distinguish the ages that is more affected. So the main intestinal bacteria identified are pathogenic bacteria like Klebsiella pneumoniae, Salmonella as well, and Escherichia coli. So we have until species. About rotavirus, we obtain the expression of amplicon that's an example of amplicon according to VP6 genotyping. So was building. We starting to do this process in the lab in Angola. Has not this method until now. So I'm very quickly. So we are thinking about new perspectives. It's because we want the accuracy results, and we want to collect, to make a collection for a biobank. So we will take the same samples and we will aliquot that. So we want to do the molecular approach for bacteria. We are not doing yet. We have not conditions for that. Also for sequencing, we maybe with collaboration it is possible. So with the cultures, we want to isolate after identification by VTEC machine. We take some pure colonies and keep at negative 80 Celsius degrees. So we want to build a biobank. So I think that's important to think about new markers for diagnosis or to develop vaccines possibility. So we want profiling also the antibiotic susceptibility. It's possible because we using the VTEC cards for sensitivity tests for the susceptibility for antibiotics. Several antibiotics are used as a marker. So in conclusion, CISA have been established a local capacity for surveillance of intestinal bacteria. We achieved the lab capacity to perform the protocol for genotyping of rotavirus first time in Angola. So a terrible prospect to expand our studies in order to accrue our results and to make a good collection of this area. So I want to present my lab team, and thanks to Dr. Miguel Brito, our principal investigator, for technicians Maria de Fátima, Analtina, Lucas, and the PhD student Sandra Miranda. So thanks to BFA, Fundação Carlos Gulbenkian, and the Cooperação Portuguesa for the funding for that allow we start this study. So that's all. Questions, please.
Thank you, Elsa, for your nice presentation. So now I think Karina has a question.
Thank you very much, Elsa, for the very clear presentation. So I was wondering if I understood correctly, when you are sampling for intestinal bacteria, you are mainly using the McConkey red agar media, right?
Right, a selective medium that select Shigella and Salmonella, and select the negative, gram-negative bacteria. So we think that 2 or 3 medias are enough to identify about 11 species of main intestinal bacteria.
Yes, and I'm curious because you also see Klebsiella pneumonia, and now I was thinking that maybe in the future we can discuss this in more detail. That perhaps when you are isolating, when you are using media specific for Klebsiella pneumonia, you might also be selecting for other Klebsiella bacteria that might not be pathogenic as the ones I talked about yesterday. There's also other species like Oxytoca, which I think might have a protection ability. They can also be dangerous if they become antibiotic resistant. But I think in the future it would be nice to discuss if you are also at the same time interested in collecting some of the non-pathogenic that can then be used as members, as storaging members of the microbiota.
I hope so. We have no access to analyze about the strains, about the right strains or accuracy for
at the strain level.
At the strain level. So since also we collected the stool, the water stool from diarrhea cases, we think that the identified bacteria are pathogenic, but I agree with you that it's maybe wrong, because the role
not wrong, but it can be, you can do more with it. Yeah.
Yeah.
And one of the challenges is still the storage.
Yes, yes, it is. Thank you so much.
Yeah.